Of course, this is the March 13th, 2021 NMGS program from 11 till noon today. We are the New Mexico Genealogical Society, and I am Robert J. C. Baca, president of NMGS. Our former president, Henrietta Martinez Christmas, is also on here. And we also have Jordan Jones moderating for us. Miguel Torres, of course, is our speaker. And let's see. Okay, Miguel A. Torres is doing My DNA Story. That is the program for today. A few things I want to go over. Our next monthly program will be in May, okay? We'll have more information about that later. Albuquerque Genealogical Society is offering NMGS members free admission to their April conferences. Again, we'll have more information about that. I want you to save the date. Our NMGS will have its 2021 virtual conference on August 27th and 28th. It's going to be a great conference. We're putting it together. We have a really good committee that is getting it all together, and we're going to have some fantastic things. So check out our website, www.nmgs.org, or just nmgs.org. Or if you're a member, you may receive an email newsletter. If you're also a member, in June, you should get our journal, and there should be more information about our conference at that time. And just a reminder, don't forget to turn your clocks forward 1 hour tomorrow, okay? If you're in Arizona, don't worry about it. But if you're in a state that actually does change its clocks, please remember to change it at that time. All right, so let me go ahead and introduce our speaker today. Oh no, actually a few things. You may ask Miguel some questions in our Q&A. To minimize distraction, all attendees of the program will be muted, but Miguel would love to answer questions during and after the program. So there on the bottom of your screen, if you're on a computer, I'm not sure how the cell phone is going to show up, but on the bottom of the screen, you should have a thing that says Q&A. It has two little speaker boxes, kind of like cartoon boxes, okay? You can put a question in there and all that. However, all questions will be monitored by our Zoom administrator, and please realize Miguel may not be able to answer all of your questions. So we apologize if we are unable to get to your question, but we hope you enjoy this program. Oh, one other thing I would like to mention is that this is being recorded. So just keep that in mind. And so it will be on YouTube at a later date. And so if you want to review this or you want to share it with your friends, it'll be there. We'll have the information on the website where to go to find this, okay? And so let me go ahead and introduce Miguel. Miguel A. Torres is a research technologist at the Los Alamos National Laboratory working in material science. He earned his BS in environmental science and an AA in Southwestern studies, both at Northwestern New Mexico College. Torres serves as administrator of the New Mexico Genealogical Society DNA Project, also known as the NMGS DNA Project, as a member of the Manitos Project. Miguel has published many genetic genealogy articles and contributed a chapter to Nación Genezaria. My Spanish is not the best, but I hope I pronounced that correct, Miguel. And more can oh boy, I wrote that down. More can be found on Miguel on his blog, the New Mexico Genetic Genealogy, and the NMGS website under the DNA tab. Again, we welcome you to look at our website, nmgs.org. You'll find more information. Without further ado, Miguel, why don't we start your program?
Good morning to everybody. Can you hear me? All right. Okay, let's get started. I've got a chat open here. Let me get rid of that. And okay, so what we're going to do today is, I was asked maybe last month, early February, if I would be willing to give a presentation, and I said yeah, and I thought, well, what do I want to do? And I said, I'm going to challenge myself a bit because I want to show what the usefulness of our DNA project is and how others, can apply what we've been building and apply it to your own, histories, genealogies, etc. So I said, this is a personal interest. So this is kind of, put me through a challenge, but it also, gave me time to reflect on what I've been doing and apply it to myself. And so I said, I'm going to tell my DNA story, basically a personal lens through DNA, through genetic genealogy, obviously. So let's get going here. So there are 3 different types of DNA tests that a person can do, right? We've discussed this many times, right? One of the most common tests is a autosomal DNA, which gives you an ethnic inheritance percentage. It also links you to known relatives based on common DNA. There's Y-DNA, which only males test, and that traces directly your paternal lineage. Only males can test for that. And then there's mitochondrial DNA, also known as mtDNA. That traces a person, both male and female, through their maternal lineage, their mother's 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 mother. And both of these spectrums, there's basically 2 spectrums we're talking about here, all the way back in time. Here for this project, we try to focus that and limit ourselves to, to colonial New Mexico. Obviously, our DNA and our genealogies go back in time, but we try to make relevance through genealogy. And although many of our families have roots to the 17, 16, and 1500s in what we call the New World, it's very, very hard to trace many of our branches that far back. We do have luxury of tracing, quite a few, but we descend from thousands of people, and so it's not always that easy. So it's quite interesting to know that we have all of this inheritance, and it's again quite hard to trace genealogically and validate this stuff. So this slide here shows my ethnic inheritance, and I thought this is a good way to show this and kind of describe this. It says that I am 63% European, and then it breaks this down. So for those of you who have tested this type of test, you obviously get this and have seen your results. We've got the Americas, which is in reference to our Native American inheritance. This says here that I'm 30%. And then we break it down further. We've got Middle Eastern and North African roots and African roots. And within those main groups are subgroups of populations. And this is kind of what tells us what we on a personal level have inherited from our broader genealogy. So if you look here, you probably can't see this, and I'll explain this further. You'll have a zoomed in version of this later, is just, a 1-page pedigree on myself, starting with me, my father, his father, his father, and his father, which is in relationship to my Y-DNA. Over here, we see to the left, my Y-DNA haplogroup, which is a population haplogroup, means that I descend from a particular group of people in relationship to migration patterns, etc. In history, my haplogroup is IP37. Well, I've tested the big Y, so that's been refined to IY32624. A little more on that is specifically with big Y analysis, originates out of the Greece region. I have theories on that. I won't go into it at the moment. My mitochondrial DNA is haplogroup A2H1. So if we look at myself again, we do the opposite spectrum. Here's my mother, her mother, her mother, her mother. And that haplogroup A2H1 genealogically is validated to descend from Maria de la Cruz. We'll talk about that in a second. I should say that the Y-DNA is validated to descend from Cristobal de Torres, who can be traced to the late 1600s. Maria de la Cruz is an Oñate era colonist. So it kind of gives you a little bit of a idea of the 2 timelines that those 2 branches can be traced to. Well, I should say, when we think about this ethnic inheritance, right, I have all these different percentages that I've inherited. Well, those are basically a result of all this stuff in the middle of my tree and beyond. right? I've got great grandmothers and great grandfathers all spread out through each one of my ancestors, right? And then that is exponential as you go back in generations. It's exponential. So it's interesting to see. Very important to note, and I'm talking to people who are very well aware of this, is that we in New Mexico descend from what we would call a limited population. So many of us have have the same ancestors multiple times over. Here is a expansion of what my father's side of the family tree looks like. So we have here, and I'm trying to move some stuff on my screen here. Give me a second. Okay, so here in the bottom right, I said, okay, let me focus because my family tree is large, and I could really talk about all kinds of branches and things, but let's just start fundamentally. And most people should be kind of working towards this to apply this to their own family trees, right? We have here on the lower left, this is my dad, Fernando, and then his parents, Floraido, also he went by Alfred, and my grandmother Pearl, she's 85, 86, and she's still alive. And then her parents, Leonor Romero and her mother, Isabel Trujillo. On the other side, Floraido's, Alfredo's parents are Alfredo Torres Sr. and Adelina Medina. Now, since I'm not going to give you the whole snapshot of that genealogy, I can tell you that from my great grandfather Alfredo, his line, when we break down him, right, he's got parents and grandparents and great grandparents, and some of the names that come from that side of the family are Muniz, Romeros, Pais, Maes, Holguin, and then on my great grandmother Adelina, she's got Vigiles, Mondragons, Trujillos, Valdezes, and Alfredo Torres was born in Talpa, and probably around the 1920s he migrated to Chacon and Mora. So just to give you a little bit of perspective as to where these families were, because what I want you to do is kind of get I'm telling you my personal story, and I want you to kind of say, hey, I have family from here or there, and I probably have these same relationships. Well, Adelina's family was in Chacon for a few generations, but had come out of the Peñasco region. Now, when we switch over here to the Romeros and the Trujillos were also my grandmother Pearl was born and raised in Lidu, and her parents and grandparents were there for a generation or two. Some of them had come from the greater Santa Cruz Valley, others from the Peñasco, La Canoa, which is near present-day Velarde. So, what I like to show you here is that, within just a few generations, I've got like all these different connections to different communities. And, that's just a small perspective of my father's side, right? And that really gets further back in time. And when we then move over to my mother's side, this is my mother, Jackie, and she's the daughter of Fares Weibal and Isabel Madrid. Isabel Madrid, her mother, was the daughter of Alberto Madrid and Rose Rodriguez. These are families from the La Vega de los Vigiles, which is basically the Espanola Valley. Rose was the daughter of Demetrio Rodriguez and Bersabel Vigil. Bersabel Vigil is the daughter of Jose Agapito Vigil and Octaviana Vigil. Octaviana is the daughter of Jose Dolores Vigil and Josefa Martin. These families had a long history and presence in the La Vega de los Vigiles area. And to give you perspective, right, it's beautiful to be able to see, a visual of this, my mother, grandmother, etc. And as I mentioned earlier, this lineage traces to Maria de la Cruz. And if you look at that and now put timelines in perspective, right, my mom is born in 58, we got 36, my grandmother in 1902. And to the furthest right, that's the furthest picture I have on this lineage. And Octaviana was born in 1858. So I just love to show this. And even with my own kids, I'm like, just take a look at that. And it's really interesting to see how a lot of our cousins still hold these features of Octaviana in their looks today. Well, I put this little note on the bottom, and I mentioned earlier that we come from a small population of people. Well, in my database, I ran a little calculation, and it said that I descend from Maria de la Cruz at least 6 times. I'm pretty confident I have many more connections to Maria de la Cruz. It's just that I haven't made those genealogical connections solidly, right? But what we've done here is that my maternal DNA has been validated not only by myself, but by others on the DNA project, right? I said, okay, I'm going to test this on myself. And I supplied the information probably to Henrietta. She double checked it and proved this. So, that's one perspective of how I've been able to use DNA. But like I said, what's interesting is that I have this multiple times over. So when I see other branches that fall within my tree that also descend, I'm like, well, I don't only have this A2H1 haplogroup one time, right? That's built into me genetically multiple times over. So that's the real takeaway.
Miguel,
Yes.
I've got a question from someone.
Sure.
Someone named Ken Pino or Pino, I don't see an Enya, but might be, asks, would taking the big Y DNA be useful? He took the Y67. So back to the Y DNA stuff.
Right. So I'll be honest with you. I've worked with Big Y, obviously, for myself and some other projects, but I don't focus on it all that much. But it is useful. It's not always most useful for genealogical purposes, but what it really does is, and it depends on what family clan you fall into, right? It's very, very dependent. It's a process. So it will help you better understand where the origin of your family is back beyond genealogy, way back in time, and how you broke down and descended, broke out from major clans in history. That's what it really helps you do. If you want to use it for genealogy, you have to really invest a lot of money and a lot of time, because it becomes a process of getting other known, validated lineages tested to really start breaking down the intricacies of that. And I really, if you refer back to last year, in March 2020 and June of 2020, the New Mexico genealogist journals had two articles, part 1 and 2 of the Espinosa case study that myself and Lee Martinez wrote. In there, it talks a lot about the Y-DNA. So I would really recommend reading those, probably more so the journal in June, because it kind of goes about what you really need to do to make Y-DNA useful. So that's a short answer, but it's very, very dependent on what you want to learn from it. And we can further discuss that. Now, going back to my maternal DNA, I just wanted to give you a real quick perspective because you couldn't really see the details earlier. So I just talked about Octaviana Vigil. Well, she's down here in the lower left. And real quickly, she's the daughter of Josefa Martin and Jose Dolores Vigil. She's the daughter of Ignacia Montoya and Ventura Martin. She's the daughter of Maria Dolores Garcia de Noriega and Juan Francisco Dionisio Montoya. She's the daughter of Maria Gertrudis Sandoval and Jose Garcia de Noriega. Guaranteed, these people populated like crazy. So there's so many Nuevo Mexicanos that have this in their genealogies. She's the daughter of Josefa Durani Chavez and Antonio Sandoval. She's the daughter of Juana Baca and Francisco Javier Durani Chavez. She's the daughter of, let me make sure I made this correctly, Ana Moreno de Lara and Cristobal Baca. She's the daughter of Catalina Vasquez, and she's the daughter of Bernardina Vasquez. That's a very popular name. Everybody, lots of people have that in their genealogies. And she's the daughter of a Bustillo woman and Francisco Vasquez, and she is the daughter of Maria de la Cruz. So this is really interesting, because from me to Maria de la Cruz, I want to say it's something in the order of like 16 or 19 generations, and that's quite long, right? As where like my paternal ancestry is maybe 11 generations, I forget at the moment. But others can trace to some of these early ancestors with only 8, 10 generations. So there's huge differences in generational links here. So just a tidbit of information. Now, my father's line has been tested to and validated to Cristobal de Torres. That Y-DNA haplogroup at just like the Y37, 67 level without any big Y is IP37. Big Y is Y32, 624. Again, going back to this, my dad is the son of Alfredo and Pearl Romero. He's the son of Alfredo Torres Sr. and Adelina Medina. He's the son of Jose Guadalupe Torres and Maximiana Vigil. And he's the son of Jose Francisco Vigil and Maria de Jesus Cordova. This Torres family went from Francisco back to Marcial de Torres, which is a few, like 3 or 4 generations, was pretty heavily, situated in Ranchos de Taos and Talpa for many generations. And then prior to that, there's some migrations into Santa Cruz, Rio de Chama, and these Torreses are the same Torres families that are located down in Bernalillo and Belen. So, that's the Rio Bajo, Rio Arriba connections. So, in theory, a lot of these Torreses that are down south can know more about their DNA because of the stuff that I have up north, right? And here we have a snapshot of the New Mexico genealogist article back in 2016 that I wrote on the Y-DNA of Cristobal de Torres. There's a little bit more of a blown up perspective of that genealogy. We've got Francisco being the son of Jose Antonio Torres, Isabel Fernandez. He's the son of Antonio Jose Torres and Nicolasa Sandoval. He's the son of Marcial de Torres and Maria de la Cruz Martin. He's the son of Diego and Rosa Varela Jaramillo. He's the son of Cristobal de Torres, and so on. I urge anybody who has, and you probably have done so because for those of you who follow the publications, you know very well about these articles. Real quickly, I'll throw, my DNA through the paternal line, and I've said this many times at other lectures, is it links to Greece. Because the Torres lineage has basically a roadblock in the late 1600s, and I also match a group of Griegos, I have a theory that maybe Cristobal de Torres was actually the son of a Griego. And why do I say that? I think because of that link, that solid link to Greece, there is that possibility. So one way that I've been trying to attack this problem is to get other Griegos throughout New Mexico to test to see if we can build a solid understanding of who the Griegos were, and then maybe compare what I have and my results and these other Griegos that I matched to that larger study. We haven't got there just yet.
Miguel?
Yes.
There's a question about the name change from Torres with an S to Torres with a Z.
Right. That's a good question. Yeah, I can answer that easily. So my name is spelled with a Z, and my grandfather, go back here a couple generations, you'll see this here. And interestingly, the picture for Alfredo Torres Jr., he's in his military outfit, right? When the story that I heard from his wife, my grandmother, was that when he went to the Korean War, he just happened to be in Texas, and there was also another Alfredo Torres, also spelled with an S, in the same company or something of that sort. And they said, we got to differentiate here. We got to know who's who. So I guess my grandfather volunteered to use the Z. That's the story I heard. But his father, Alfredo Torres Sr., interchanged Z and S. I've got documents where he actually signed with an S, others where he signed with a Z. So it's pretty interesting. But from that point back, it seems that it's consistently Torres with an S. And so there's no real solid understanding. There's been stories, and I hear this with other names, with Espinosa, Gonzalez, Torres, whatever. People say, oh, if you have a Z, you're of this origin, more of Spain or Mexico or vice versa. There's, in my opinion, there's no truth to that. It's just early 1900s, there was this shift in spelling. That's kind of the simple answer. So here, now we expand my genealogy, right? This is my dad's mother. And I like to use this because it's really, I've presented this before. My dad's maternal DNA is haplogroup X2A1B, which is a minority in New Mexico. There's only like 3 people that have this DNA, and they all match. It's my father and 2 others. Well, the 2 other, we haven't made the connections genealogically, but the DNA is there, and they all have similar genealogical dead ends. The other, one of the others traces to the same time period as my dad, but we can't make the genealogy connection. The other person has a roadblock like in the 1850s or 60s, so we're not sure where that person actually connects to us. But what my dad does have is he has mtDNA matches to people who still claim to be of the Ojibwe nation. So that leads me to believe, if we look at her my dad's genealogy, this is his mother. She's the daughter of Isabel and Leonor Romero. She's the daughter of Ambrosita Arts and Jose Presentacion Trujillo. And she's the daughter of Margarita Romero and Julian Arts. She's the daughter of Benina Vigil and Benito Romero. These are all Lidu people until we get back to like Margarita, to Benina. Benina's like from La Canoa. And then her mother's Manuela Sanchez and Antonio Jose Vigil. Her mother's Ignacia Villapando and Diego Sanchez. Interestingly, I have this gut feeling because of the timeline, and there may be, I think this Ignacio might have used the Martin name. I have a feeling this might have some relationship to the Villapando and Hacienda in Taos, and may have some correlation to the Comanche raids in 1760. Just a theory. There's no proof to it, but how did this Ojibwe DNA get into New Mexico? And it's obviously not in high frequency. It's super rare. And
Miguel, I have a question. So
Sure.
This is your dad's mitochondrial?
Yep, my dad's
Okay, so that would be your dad's mother,
Yeah,
as opposed to the mitochondrial that you did off of yourself. So it's a different
exactly.
line. It's a different line.
Yep. So my
wanna...
mitochondrial is tracing me to Maria de la Cruz, but through my father, which I inherit some of that DNA, but not in the mitochondrial form, right? And so through my dad's mitochondrial DNA, this is the lineage and the result. And it goes back to that one of those early slides where I said, let's focus on my 4 grandparents on each side. And this is one of them, right?
So Miguel, there's a couple of questions I've got here.
Sure.
So Eloy Martinez has a question and kind of a follow-up. So he says, do you say the DNA is there because there's an overlap on a specific chromosome? And also pointing out, just because you're in the same haplogroup doesn't mean that you're related, because there's a lot of people in that haplogroup, but it goes back beyond genealogy timeframes.
Right. So to answer that question, and I'm not sure exactly specifically what they're talking to, but I'm assuming it's related to the Torres Griego connection. And the reason I say that is, yes, many families throughout the world, but specifically New Mexico, there's many families in New Mexico that have common haplogroups. RM269 is one easily to talk about. That's a Western European haplogroup. Families like Martin Serranos have that. The Medinas, there's clans of Medinas, there's clans of, all sorts of different families, Maes, there's all sorts of families, and they have the same haplogroup, which means they come from a larger population in the world. But the way we differentiate them is they have different STR sequences. Basically, when you do your DNA, you have all these numbers, right? So let's just put this into perspective. The Garcia de Noriega is a haplogroup I. The Torreses are a haplogroup I. That we're IP37, they're IM253. We come from a similar population in the world, but the way we differentiate ourselves is that we start looking at the sequences, and that breaks us down into different subgroups of people. So when I said that the Torreses and the Griegos match, I actually have 3 or 4 different Griegos that actually match my DNA sequence perfectly. So the answer to that is that either those Griegos are the true Griegos and I'm a Griego, or that there's a break in that Griego branch, those branches, and that they're really Torreses, because it's not just haplogroup, it's exact or very closely to exact STR matches. I hope that makes sense. And I can show you that when we get to the website. I'll explain that a little better if we don't forget about it.
And that was the question they were asking was are you seeing an overlap on a specific chromosome? And you are.
Yeah, exactly. As opposed to just because if we were to say that, if you look at the database, we might have 50 different families and that progenitors that have RM269. Well, they're not all identical. It just means that they come from the same population group in the world.
Yeah.
Okay. So, yep. So here is now shifting gears to my mother's side of the family. My mother's grandfather. So let me go back a bit just to give you a little perspective. So my mother's mother is here, Isabel Madrid. Her father is Alberto Madrid, and Alberto is the son of Jose de la Luz. So now, and right here, let's look at this. There's my mother's mother, Isabel. Her father is Jose Isabel Alberto Madrid. Now we go forward to this picture. These are his parents. Okay, so in order to get the DNA of this family, I had to test cousins. So I tested a male Madrid cousin who gave me a perspective of the paternal line that traces through Jose de la Luz, and that result, well, Jose de la Luz his father is Anastasio Madrid, here from Santa Cruz Plaza, and Rosalia Lopez was the mother. Well, Anastasio was in his we can't find a birth record, but we find a marriage record, and it says he's the illegitimate son of Secundina Madrid. So they're right off the bat, we don't know who his father is. So I'm going to suspect that he's not a true Madrid paternally. And the Y-DNA tested for this line resulted matching Lucero de Godoy. So he somehow, in some fashion, we haven't proven who his father is, but I know his ancestral progenitor in New Mexico is going to be of the Lucero clan. Now, again, how do I know it's Lucero? Well, just look at our journal articles. There's a whole article on the Lucero DNA, and he matches that DNA. So we have to take what we learn. It's just not that he matches a person. He's matching something that we've studied in depth. Okay. Now, Emiliana Martinez, I have this. It was very interesting because we have an oral story that she had. They, quote unquote, said she had a lot of Navajo blood, right? And we have, most of us have these stories, right? Oh, we have, we had a grandmother who was Native American, or she this, that, or the other thing. But her story was that she had a lot of Navajo blood. Well, it was very hard, but most recently I was able to acquire mtDNA from a cousin who traces through her. Interestingly, that DNA resulted in haplogroup B2. Well, B2 doesn't mean it wasn't Navajo or she was a Navajo descendant, but from a frequency of statistical frequency, haplogroup B2 occurs in Pueblo populations quite frequently. Doesn't mean that she was Pueblo, but anyways, that's just a little tidbit. Well, what's more interesting to me is that she doesn't have any matches. So if this family line, in my opinion, would trace deeper into colonial New Mexico, I would assume, just based on the large database we've built, that she would match somebody because she would have some common ancestor with somebody already tested. Well, she doesn't. And so that leads me to believe that this story of her being or having a lot of Navajo blood, meaning that she was probably herself or her mother, grandmother, she had a maternal DNA connection to being Native American in what we would call a more recent past, right? So that's a tricky subject, but I just wanted to kind of throw that out there. So when we look at DNA, right, in Jose de la Luz's case, he doesn't have a father listed, but we can connect him to a family that's well established. Emiliana is not being connected to something that's well established, which makes me lean more to her being of a more recent Native American origin. However, what's strikingly interesting to me, and I'm getting personal on this level, right, is that she, to the right, are her parents, Ismael Martinez and Manuela de Atocha Montoya. So her mother, she acquired her mtDNA from Manuela de Atocha. I don't know if I trust the way these pictures look. She doesn't strike me as looking like either of those two parents, but her baptism and her marriage all list her as being a legitimate child of this couple. So I've got to take that record and believe it, right? But it's striking that even physical features don't really show her. So was she Native American? Maybe she was adopted and legitimized. It's something to be questioned. Well, now, taking this perspective even further, right, I don't have the ability to test for the Y-DNA of Ismael, her father, but his genealogy is validated to descend of the main Martin, they are non-Martin Serrano clan, specifically through Blas Martin Serrano. And, we have an article back in 2016, and Jose Antonio Esquivel has done some excellent work, Henrietta Christmas, on the Martin Serrano people. And so the genealogy is very well validated. Although we haven't tested a cousin that's a Martinez, because the genealogy is solid, I would assume that my Martinez connection via Ismael, is legitimately, and I would acquire that same DNA, if that makes sense. Now, the caveat to that is that if there is an illegitimacy or something in the records something not reflected in the records, and there's a break in his genealogy, therefore he wouldn't be a real Martin Serrano. That I can't say. But based on the genealogies I have to compare, I trust that he's a legitimate descendant. So now let's switch gears here to a little bit of here's a little self-promo. So the book that Robert mentioned earlier, in Nación Genízara, is a wonderful book that was published, a year and a half ago. And it's all about the ethnogenesis, place, and identity in New Mexico. I had the privilege of writing a chapter on what we're learning about DNA and in New Mexico, and more specifically, what we're learning about our Native American DNA through genetic genealogy. Well, the page that you're seeing here, you can obviously read it, but what I did is I showed a real short case study on an mtDNA link to many new Mexicans to this person who is of full-blooded Native American origin. And to the right here is a picture of his grandmother or great-grandmother, Amelia George, who was born somewhere in the mid 1800s. That genealogy is stuck, but she was born out in the Navajo, I think, Red Rock region. Her oral history is that her family, the oral history in that family is that they migrated from this Tewa region, right? So I think a little bit more in depth, and those of us who have studied, migration patterns, we can, and history of New Mexico, we know that the Tano people of the Santa Cruz region migrated out to Hopi lands, and a lot of those Hopi people were raided by Navajos, or willingly after the Pueblo Revolt or during the Pueblo Revolt migrated into Navajo, had a lot of Navajo interrelations. So it's just interesting that his maternal DNA links to many Nuevo Mexicanos, specifically of the northern New Mexico region. And here's a picture of a woman who's from Truchas that matches that lineage. Well, this lineage descends from Barbara Anaya. And when I did this work, many of us were stuck with Barbara Anaya. And when we did the ABIQ DNA project, Henrietta was able to take some information and expand Barbara Anaya. So here you see Barbara Anaya was married to Antonio Jose Lopez. She's the daughter of Feliciana Gertrudis Ulibarri. She's the daughter of Josefa Garcia de Noriega and Alonso Rael de Aguilar. So many people in New Mexico have these ancestors. And Josefa Garcia is the daughter of Teresa Varela and Alonso Garcia, again, founding families. Well, this Teresa was the daughter of somebody, right? And we assume it must have been a Varela woman. But so what this is telling us is that we have validated genealogies back to this mid to early 1600s era, which is pretty consistent with these migration stories of this family that I just mentioned. So what does this do for us? It tells us that, hey, we have this maternal DNA. So like I said, many of us have these 4 women in our genealogies. I have Barbara Anaya many times over. I didn't present that to you today, but I did a calculation and she appears multiple times in my genealogy. So this is one way that we can take something and apply it to our genealogy. So the haplogroup for this, I believe, was a B2 something, or a haplogroup B. So we can then apply that haplogroup to many branches within our family tree. From that same book, this is the cover page, the helix. The title of the chapter was "The Helix of Our Native American Ancestry, Native American Genetic History." I urge you, this is an amazing book for learning about our Native American history, not only through DNA, but it touches on so many topics. Now, this picture here is of my grandpa, my mom's grandfather that I was mentioned earlier, Alberto Madrid and Rose Rodriguez. I didn't really say much about her, but we've tested her side of the family as well. She's a Rodriguez. Her paternal side traces her to Fray Jose de Castro, who was the last Franciscan priest in New Mexico and had relations with Ignacio Rodriguez and had many children, one being the branch that I descend from. So I have this paternal link to a priest in New Mexico. And on the opposite spectrum, her mother's side, which I did show a little bit of because she was in that first mtDNA line, but that side of the family is full of Vigiles and Martinezes. And so there again, I can apply all the DNA that we've learned about the Vigiles and the Martinezes to that side of the family. So like I mentioned before, this thing gets really exponential. So one of the tasks that I have at hand, and I will get to at some point in my life, hopefully, is to be able to build this family tree and really just mark down all of these validated haplogroups and how they fit within my tree, because then that is a reflection of how I got that ethnic inheritance. So hopefully you're following along with that. I'm going to stop sharing that screen. And while I migrate, if you have any questions, I'll entertain those. So what I would like to show you is, let's navigate. And I think we're probably running out of time real quickly here. So I wanted to real quickly navigate to how you all can take information that we've built for you and start to apply it to yourself, right? That's the whole goal of what I'm trying to show you. I'm showing you my personal perspective. Let's apply it to you. So are we seeing the NMGS DNA project page?
Yes.
Okay, good.
Yes.
So when you get to this, so now you're basically seeing my internet here. So there's several ways to get to a lot of this stuff. The easiest is let's just go to whenever you search this stuff, if you just go to nmgs.org, you're going to have all these tabs, informative tabs. Well, if you click on the NMGS DNA project tab, you're going to get a whole bunch of really good information. Who's helping us validate this stuff? What are the different types of tests? What are they doing? What are our project goals? And links to this, all the different things, to our FTDNA page, FAQs, the project, forms that we require, my blog, issues on DNA. So everything that has been published since 2015 to the present is listed here. So if you want, here on these articles, if you want, there's downloadable one on this tab. If you want to figure out, well, what articles have been published, you can go to this link and request from NMGS to purchase those and get those if you don't already have them. Well, if we were to click this tab, it basically takes us here to this. This is where you join us. Well, an interesting thing here you need to do is to navigate basically to these DNA results. If you click DNA results, it takes you to this page that we were just on. You got Y-DNA and mtDNA results. Let's just take a quick look at DNA result, mtDNA. When you click this, it's going to open a page that has our results. It's currently set to 25 names. I just like to just increase this to 1,500, try to capture everything possible in our project so that you can do it all and you don't have to migrate page by page. So what you can do here is you can scroll this page, and what it does is for the participants, it lists the kit number, their last name, the last name of the person tested, and then who they claim is their last maternal DNA ancestor. Well, our project has migrated to this method of whenever we validate a lineage, we update that last known ancestor to include an asterisk. So it gives myself and the public it says, okay, looking at this page, I know that this participant's mtDNA has been validated to this time period. Well, in this one, it ends in 1842, but you can do this all the way down. I guess it didn't take because I asked this to go to 1,500, but in any case, otherwise you have to do it page by page. Well, let's just go to page 5 as a test. You can do a find function. And so let's just say that here's all these different kit numbers that trace to Maria de la Cruz. If you have, let's just say that you have a Maestas ancestor or Mestas, and you want to see, does this database have Mestas? You can do a search and find, and I don't see it there, but in any case, if you wanted to know more about Fernandez, you could say, okay, hey, Miguel, you would write to me and say, hey, kit number 40621, let's just use this as an example. Hey, kit number 631363 has a validated genealogy to Maria Jeronima Chavez. Well, you're not getting her full genealogy or who she's married to, but you say, hey, I also have a woman with that same name in my genealogy and of that time period. I want to compare and see if that's the same woman. You would email me and tell me this information, and I could probably share a little bit about who this person was. That's kind of the usefulness of it. That same perspective applies to Y-DNA. When you get to this page, again, you just search, and you can just do a search for an asterisk, and you'll start getting all these that are validated. It's really, really cool and interesting right here. As an example, here's Juan Jose Bustos, born in 1722. It's a validated lineage. If you share him as an ancestor, you know that his results are haplogroup G-M201. And our project is very specific that we validate to the last known ancestor. So in some cases, they may end here, like Miguel Lovato ends in 1847. But, is he or is he not a true descendant of Lovato? What we do is we compare him to his matches, and he might really be, this is, don't quote me on this, right? He might really be a descendant of the Archeveques or the Silvas, depending on where he falls in that STR sequence. Anyways, I think I'm talking a little fast because I see the time is running out, and I'm happy to migrate and navigate this database more. but I think it's probably a good time to end and to get some questions, because I think I can make use of this a little bit more.
Miguel, can you hear me?
Yes, I can.
So someone asked about the ABIQ DNA project, and I emailed them back, or I chatted in the box with them about the journal that was published with the findings. I don't know if you have anything else to add to that.
Well, I can say that when we did the DNA project in ABIQ, we were specific and we studied, 10 men and 10 women. And so, but in our DNA, so that's specific. So the article talks about those, those 10 people. Well, our project here, the DNA database, actually has many, many, many more people here validated and traceable to ABIQ as well that are not reflected in the article. So I think when I did the Y-DNA, if I can get here, an example of that is, let's see. So an example is there's a large Madrid family that still lives in the Rio Chama area, all the way up to Chama and Tierra Maria. And most of them are traceable to, his name was Salvador Horta Madrid. Well, he's the son of Miguel Pacheco, who in the records is is listed as an Apache. And the DNA is a haplogroup C, which is consistent with Athabascan DNA. So my point being is that our database here has tons of other lineages validated to ABIQ, as well as let's just talk about other areas, right? Validated to many communities in New Mexico. We can start organizing this by communities and say, okay, we have all these lineages traceable to Trampas, to Talpa, to ABIQ, to Santa Fe, to Bernalillo, to Carnuel. It's pretty interesting. So that's kind of, again, the ABIQ project is like a case study on exactly what I'm talking about, how we can apply it to our trees. Hope that answers the question.
Thank you. We have another question in the chat box. A woman says that she has her brother's Y-DNA and has contacted the people and has gotten no response. So what kind of information can you share from their kit? And she's not sure if they're part of the NMGS project.
Yeah, so if the person that's being questioned, the brother, if he belongs to our project, then I would have the ability to go in and see who his matches are. If I looked at those matches and I say, hey, some of these matches I know I can recognize as being our participants, then I could help this person by saying what we know about those specific lines. But what this person is describing is something that's a common thread and has been since the inception of our project and beyond, since we first started doing genetic genealogy. Many folks just are reluctant to respond to our requests. There's many reasons for it. Sometimes the people that are doing DNA testing are not doing genealogy. Sometimes they just don't understand the technology. There's so many reasons, but I would say the best approach is basically you reach out to me if it's a member of the project, then I can tell you, hey, here's who we do or don't have in the project that we can help you with in relationship to your line. And if none of them are, then you're kind of stuck with, either continuing to try and probe them, or it just may be a dead end.
So another one question is, what do the different colors mean for haplogroup, red versus green?
That is, I don't know that answer off the top of my head. I used to know that. I think I'm pretty certain what that means is that there's specific SNPs, locations on the DNA that when they're tested, they confirm the haplogroup. So people may go into their results and it'll say your predicted haplogroup is QM242. It says predicted. If it's confirmed based on some specific marker, it turns green. I'm pretty certain that's what that means, is that we have confirmed haplogroups versus non-confirmed. But most cases, these non-confirmed, all it takes is some specific test to confirm it. And it would be like buying an SNP package to validate that. And those are questions that, we can answer on case-by-case basis. Well, real quickly, if you look at up here, I'll just add to that. You see all these DY markers. These are the locations on your DNA that are, extracted and put into numerical order. Well, these that are, so for anybody up here, or for anybody looking at this information, whenever you look at your results and you see these that have the red columns mean those are markers or genetic locations that mutate faster. So if you look down and you have matches to people and you say, well, I'm a genetic distance of 3, and all 3 differences land up on these red columns, it's just because those are locations that mutate faster. Because I've had people make the argument, well, I'm not an exact match to that person because I'm a difference of 3. And it's not that you're totally different, it's just that mutations have occurred in one line or the other. I hope that makes sense.
Thank you. Another question is, what's the difference between our New Mexico DNA project and Angel Cervantes's project?
That's a really good question. So when I started doing genetic genealogy back in like 2006, I wasn't an official admin or anything of his project, but I really tried hard to recruit people to build this kind of knowledge and get them to join his project. His focus shifted over time, and he should better talk for this himself, but I can say what I know is that he's really migrated to a anthropological view of what our lineages are telling us, right? He's not as focused on validating genealogies, and he doesn't have the manpower to do it. So the advantage to our program is that we have a team of folks that are validating the genealogies and then we're making sense of what the DNA means in comparison in relationship to specific ancestors, where he may just take DNA sequencing and last names and talk about where their ancestral origin is back in Europe or Native American populations. So we're more focused on understanding the DNA and the genealogy.
Okay, thank you. I have another question. Father's DNA for the project was from Abiquiu. He was QM242. Oral history is that he was a Genízaro. Found him in the census, but he has no matches. What do you suggest she does next?
That is an interesting one. I'd be curious to know if that person is part of our project. If not, it should be. It's when you don't have, that's basically the same case that I have with my Emiliana Martinez, right? I tested a cousin and she has no matches. There's really, the only thing that I can do and would apply to this same scenario is try to look at that extended genealogy of that person. So if I took Emiliana and her mother and grandmother and tried to see if there's other branches that have living descendants today that I could test as a comparison to see if we can make common links. At the end of the day, you may still end up with, maybe it's just that it's a solid line, goes back to Abiquiu, and there's just no other descendants out there that have tested to date. That's probably, that's the reality of what we're seeing there. There's really not much more you can do about that. And I think that's actually a more desired outcome, because if you start matching families, and I'll give you a perfect example, Henrietta's been working on it with me, Angela Lewis, Daria, and several others. We're studying the Garcia de Noriegas, the Gurles, the Durani Chavez's, and there's other families within those groups, and we're studying them through Y-DNA. They all have solid genealogies, matching DNA that trace them to the late 1600s or mid 1600s or so. But we can't make sense of who is really who. Are they all the same family? Are they independent families that may have common links, earlier in Mexico or even in Spain? We just can't make sense of it yet. And so that's a really hard one. So what it does is it puts all the families that fall within that group, which are a whole branch of Medinas, Vallejos, just to name a few, right? Garcias and others. They don't, at the end of the day, we don't know who's who in that group. So that's more confusing and hard to deal with. So in this case of like not having matches, more desirable, because I think that you're a true descendant of that person. And it's just that there's not others tested for that line.
Thank you. Okay, I have another question. So someone has a brick wall that you have verified. How can that help them with their line? And I think you explained that a little bit with people going into this slide that we're looking at right now, but maybe you can elaborate just a little bit more.
Yeah, and we have tons and tons and tons of examples of that, right? and I'm just trying to see if I can find some that are really quick and easy. But at the end of the day, we have, for example, here, this is a perfect example. Like if we're looking down here at this, Miguel Garcia. So this particular genealogy has a dead end in 1800. It has an asterisk, so it means that we validated to Miguel Garcia. But he does match other Garcias that are traceable to Alonso. So this Miguel Garcia, the participant may not know how Miguel connects to the progenitor line, but can be assured that based on the DNA matching, that they are also descendant of that progenitor line. And that's not the best example because we're talking about this whole Garcia Chavez scenario, but that's basically the approach is you have a dead end. And if you're lucky to match others who have extended genealogies beyond that time period, you can, in most cases, be assured that you're also a descendant of that same progenitor line, whatever it may be. It's just you may never be able to know how.
Thank you.
But what I should say is what it does is it, in addition, now gives you clues to where you may not have been thought of, right? So let's just say that this Montoya had a dead end in 1827, but is matching the Valdezes. Well, if this Montoya says, well, I'm matching Valdezes, I'm going to go back into the historical records, censuses and marriage records and see, are there padrinos? Are the Valdezes the neighbors? It starts to produce some clues on where to go and where to look. You may not come up with results, but it starts to direct your research.
Thank you.
Okay, we probably should start wrapping up. It's noon here in Albuquerque, and I'd like to thank Miguel again for presenting this. Very good information and all that. I would like to thank everyone who attended. We had a really good number here. We had about 108 people showing up at its height. And so again, thank you, Miguel, for your presentation. Thank you, Henrietta and Jordan, for helping out with this. And if there's anything else you want to just kind of finish off today, Miguel.
Yeah, let me finish with one final thought, and that is, I showed you today an example of how I'm applying this to my family tree, and I only, I focused on the 4 sets of grandparents on both mom and dad side, and maybe a little bit beyond, right? But that's a very small window in my family tree. Now, what I would urge is that you start applying this to your family trees in that same manner. Just start slowly and working back. In addition, you can, again, it's so awesome to just go down this list on this database and pick any one of these validated lineages, and just then go into your database and say, okay, do I also, like Olivas is a very common name in northern New Mexico. Not very common, but I mean, he's a common ancestor to many people. So all you got to do is just go into this and find these, go into your database and do a search and say, do I descend from Pedro Asensio Olivas? And if you have that connection in your tree in some manner, you can come back here and be rest assured that this validated line, in theory, should apply to you as well, and this haplogroup, with the caveat that assuming there's no illegitimacies or broken or bad genealogy in your tree.
Okay, very good. I just want to remind you that we will have this presentation later online. It's going to be on YouTube, Jordan. Is that where it's going to be?
He's muted.
Yes, it'll be on YouTube.
Okay, so keep an eye out. If you're a member, you'll get a notification in your email. If you're not a member, just go to the nmgs.org website. We'll have a link to this presentation. And again, I would like to thank everyone who participated, and keep an eye out. We will have a program again in May, and we'll put more information again online and in our email newsletter. Thank you very much.
Been a pleasure as always.